students, in this session we will see the second event of the post-fertilization event, that is the embryo development. Now, embryo development occurs at the micropylar end of the embryo sac where the zygote is present. Now, zygote, as it is present at the micropylar end, the development of zygote occurs when some amount of endosperm is formed. Now, why this happens? Because as we have discussed in the last session that endosperm, it is required for the nourishment of the developing embryo. So, zygote development and the division starts only after certain development of the endosperm. Now, what happens in the embryo development? So, here the zygote divides repeatedly and it forms many structures. First structure that is formed is known as proembryo. Second structure or second stage of embryo formation is known as globular stage. Third stage is known as heart-shaped embryo, and the last stage is known as the embryo, where that is the final stage of the embryo formation. So this is how embryo formation occurs. Now we will see how the dicot and the monocot embryo differs. First one, that is the dicotyledon embryo. What is present in the dicotyledon embryo? The dicotyledon embryo, generally it is having the two parts. One is the embryonical axis and second one it possesses two cotyledon. That's why the name it is given dicotyledon. So now above the embryonical axis, the part of this particular dicotyledon embryo is known as the epicotyle, which terminates or which particular ends at the plumule, that is also known as the stem tip. And the part below the cotyledon portion is known as hypocotyle and it ends at the radicle or the root tip. The root tip, it is again covered with the root cap. So this is all it is present in the dicotyledon embryo. Now in the monocotyledon embryo, as particular cotyledons they are not present, or you can say that only single cotyledon it is present. In most of the grass family, this cotyledon it is represented by the word scutellum, that is present at the lateral side. Now, two more things are present in the monocotyledon embryo, that is the plumule, that is the shoot tip is there, that is covered by one sheath and that is known as coleoptile, whereas the root portion or the radical portion from where the first root begins, the covering of that radical is known as coleorhiza. So these two terms you have to remember, that the covering of the radical is known as coleorhiza and the covering of the shoot tip, or you can say the plumule, is known as coleoptile. And the examples of the dicotyledon embryo, that is, which are the examples are there, that is peas, bean, they are the examples of the dicot. And monocotyledon, that is sunflower, maize, all this are the example of the monocotyledons. So this you have to remember for the embryo development.